Okay, guys, so moving into everything that we talked about last time. If you remember last time, we had discussed reactions. Basically, we got into the first and second laws of thermodynamics. We talked about first law of thermodynamics is all about energy transfer. So pretty much that, you know, we can transform energy, but we can't create new energy, or we can't destroy what's there, but we can transform it. We can transform it, for example, from sunlight energy into glucose energy, like we saw with the plant, and then a human could take in that plant and then make ATP energy from the glucose, which is what we're going to be talking about today. And we additionally talked about the second law of thermodynamics, which talked about how when we basically transform energy from one form to another, we lose a little bit of energy as we go, because some of that energy is going to be lost as heat, and heat itself is a form of energy. And when we do this, we increase disorder of the universe, or entropy, as it's called. And we need energy in some type of organized state in order to be able to do work. And that is what metabolism is all about. It's us taking a particular form of energy and this energy in an organized state and breaking it down so that we can make energy that we need, that we can use for our own cellular processes. And we talked about the idea of coupled reactions. So the first type of reaction we talked about, which was coupled, was endergonic reactions and exergonic reactions. And who can tell me what endergonic reactions do? What do they do? What do they form?
Free energy?
Well, endergonic reactions they can form free energy. They could, sure. But just not specifically, but more generally, what do they produce? They produce a product that does what, or has what? It has more energy than the reactants, remember? So we're producing a product that has more energy than reactants. So, okay, I'll write this on the board again. I did before. So if we're talking about an endergonic reaction, an example of this would be if we take maltose and maltose. Oh, I did that completely wrong. I wanted to say glucose and glucose to form maltose. Yeah, you guys. There we go. Did that completely wrong. Glucose and glucose to form maltose. That would be endergonic. Why? Why would that be endergonic?
Is it because it's creating more usable energy? Or is that exactly what I got wrong the first time I answered the question?
It's because when you combine two monosaccharides like that, you're actually creating a structure that has more bonds. Where is energy stored? Basically in bonds. When you break the bonds, you can create energy. So keep that in mind. When you're making a bigger molecule, that molecule is going to have more energy. So our reactants, the glucose and glucose, by themselves have less energy than the maltose does, which is the disaccharide. Exergonic reactions, on the other hand, exergonic are going to be the opposite. So here we're going to have products that are produced that have less energy than the reaction does. So if we took this reaction and we did it in the reverse and took maltose and took a disaccharide, broke it down into two monosaccharides, then we would have less energy in the monosaccharides than we have in the disaccharides, and that would be exergonic. So just think of it. Ender means into, so you're putting energy into your product. Exergonic, you're taking energy out. It's exiting out of your product. Okay, so that is where we pretty much left off. And this is what ATP looks like, which again, ATP is formed by what type of reaction? Adenosine triphosphate is usually formed by the reaction between adenosine diphosphate, ADP, and an inorganic phosphate. We call it inorganic because it is not attached to adenosine, which is a carbon-based molecule, which makes it organic. So we always have like a little lowercase p, a little lowercase I next to the p. So for example, ADP plus this inorganic phosphate, because it's not attached to the molecule yet, so it's not organic, creates ATP. Is this endergonic or exergonic? We go from adenosine diphosphate to adenosine triphosphate. Is that endergonic or exergonic?
Ender.
Absolutely, endergonic, right. If we take the ATP energy and we use it to do something, and we end up with ADP plus inorganic phosphate, is that endergonic or exergonic?
Exergonic.
Exergonic, right. The same idea. So here we have ADP plus phosphate, which creates ATP. So you can see that the adenosine molecule is here in the yellow and the red. The green part shows 3 phosphate groups. And notice the little squiggly bond that's here. That indicates that these are high energy bonds. And when they're broken, they ultimately you can do work with that. So ATP can lose the first phosphate to do work, to produce ADP, but it can also lose the second phosphate that it has as well and become AMP, adenosine monophosphate. But we don't usually talk about that here. The processes we talk about simply use one of the phosphates for energy, and that's it. Okay, next type of coupled reaction. So remember what coupled reactions are. They are reactions that have to happen together. So when we talk about endergonic reactions and exergonic reactions, basically for everything that we put energy into, something has to lose energy in order for that to happen. So that's why they are coupled. And we showed examples of that in the last class when we were talking about the breakdown of glucose and then the formation of ATP from that. When we break down the glucose, that's exergonic. When we make the ATP, that's endergonic. And so those reactions are coupled, they happen together. Another type of reaction that's coupled is oxidation and reduction. Now, this is a type of reaction. When we talk about oxidation and reduction, we're talking about not energy transfer like we talked about with endergonic and exergonic reactions, but we're talking about electron transfer. So the easiest way to remember what happens with an oxidation and a reduction reaction is to remember Leo, who is a lion, and he goes grr. Now, there are other ways you can remember this too. I think there's oil rig or whatever, but this is the way that my friend Mingma, Sherpa, and I think his family, they were Sherpas because he was from Nepal. And he was brilliant at chemistry because he took it like 10 times. And so he knew all the tricks. And I don't know why he took it 10 times. He didn't have to. I think he just wanted to stay in college. Because he always stopped coming the last couple of weeks of class, and then he would ultimately flunk, and then he'd have to take it again. I never understood that. I always wonder what happened to him. So anyhow, Leo stands for lose electrons in oxidation. And Gur stands for gain electrons in reduction. Now, if you try to think of it any other way, you'll get yourself confused because, of course, reduction makes you think something's getting smaller. But in fact, in this case, we're talking about gaining electrons. So that's why I'm saying, remember Leo, Gur. If you try to think of it rationally or some other way, you get yourself very confused. Okay, so Leo, lose electrons in oxidation, you gain electrons in reduction. So if a molecule gains electrons, it's reduced. If it loses electrons, it's oxidized. Now, don't get hung up on these definitions of a reducing agent and an oxidizing agent. I'm not going to get technical in talking about things in that way. So you can pretty much skip this part when you're trying to study. I'm going to give you some examples. Now, a lot of times these types of reactions involve transfer of hydrogen instead of electrons. But truthfully, hydrogens, of course, have an electron associated with them. So if you gain an electron, or if you gain a hydrogen, you're gaining an electron because the hydrogen's electron comes with it. If you lose a hydrogen, you lose an electron because the electron goes with it. Now, coenzymes, which we sort of mentioned generally in the previous lecture, we talked about cofactors and how they could work. But coenzymes, I like to think of as sort of like postal workers in the world of metabolism. They're letter carriers, and the letter they carry is H. Right? So the letter that they carry is H. So we have NAD, nicotinamide dinucleotide, I think it is. It's derived from the vitamin niacin. FAD, which I forget what that stands for actually, but FAD is derived from riboflavin. So these coenzymes help out with processes, and ultimately they carry around hydrogens. So NAD, if we had an NAD molecule that was attached to a hydrogen, we had NAD here. NAD has the ability to actually bind to two hydrogens. And it would be written as NADH. Some people write it as NADH2, but probably the most correct way is by writing NADH plus H, because, and this is again just a technical thing, usually when the hydrogen binds to NAD, one of the hydrogens binds closely to the molecule and the other one loosely binds to it. So that's why they indicate it that way. If we had FAD, FAD also can bind to two hydrogens, and it would be written as FADH2 because both hydrogens bind closely to the FAD molecule. Now, if we go, tell me which of the two forms, NAD or NADH plus H, which of these forms is the oxidized form? NAD or NADH plus H?
Plus H.
Okay, NADH plus H, all right, what has happened to that molecule? Has it gained or lost hydrogens?
Gained?
It's gained. If you gain electrons, you've reduced the molecule. So NADH plus H is a reduced form of this because...
Isn't that what I said?
Huh?
I thought that's what I said.
I think you said off. Did you say reduced?
I meant reduced if I didn't say it.
You meant reduced. Okay.
Sometimes I don't know what I say.
Well, you're on the right track, and perhaps I heard you wrong. Or maybe, no, maybe I just...
It's on video. We'll find out.
That's right. Hi there, YouTube world. Okay. Anyway, so we have so then if we look at NAD, I always forget, and it's a good thing that you're doing that because I think I get nervous. NAD then, is this the oxidized or reduced form? It's going to be oxidized. It's the oxidized form because it has less electrons, right? So you lose electrons, oxidation. So likewise, it's the same. FAD here is what? Oxidized or reduced form of FAD? Oxidized, and FADH2 is reduced. Exactly. So now you understand oxidation and reduction. Now the question is, how are they coupled? And I'm going to try to give you some examples there. Now this diagram, this is just showing us like the whole full out structure. Yes?
Can you show us how, like, which side has what amount of electrons?
Which side has?
Like, you know how you said this side has less electrons, this side has more electrons. Like, how do you know that?
How do you know that? Well, okay, hydrogen. Do you remember the atomic number for hydrogen? 1. Exactly. Atomic number tells us what? Proton number? And electron number, right. So we know that hydrogen has 1 electron. Each atom has 1 electron, right? So if we start out with a molecule of NAD, okay, it gains 2 hydrogens, it gains 2 electrons. Does that make sense?
Yeah, so 2H doesn't have any electrons? Or is the
They do, it does, but it's not connected to NAD yet.
Okay.
So we're saying
Oh, so NAD gains 1 electron?
it gains 2 because it gains 2 hydrogens, and each hydrogen has 1 electron. So I think that that's like the key thing is understanding. Here we've got NAD by itself right now, but if we combine it with the 2 hydrogens to form NADH plus H, then NAD has gained electrons in that reaction to form a different, and that's the job. And you know, NAD and FAD, they're like letter carrier. Like I said, they're like postal workers. They carry around the letter. They carry around hydrogens. When they gain the hydrogen, they're going to be reduced. They gain the electrons. When they lose the hydrogens, they're oxidized. You're welcome.
Can I ask a question too, just so I make sure I'm getting it? When you use like the first example, just the NAD without the plus 2H, that is what is oxidized.
Yeah, this is the oxidized form.
Yeah, okay, that's what I was asking.
Yeah, that's
Thanks.
oxidized form. This is the reduced form right here. So here, I'll write it for you. This is the oxidized form. And this is the reduced form. And the rest is just showing that we're gonna
Yeah,
have,
I was looking at it as a whole
no,
thing.
this is just the reaction.
I have a question.
Yes.
And then on the bottom one, how come the 2H on the other side is in H plus H?
Because over here for FADH2 is because both hydrogens tightly associate with FAD. If this is the one good thing about this particular diagram, it kind of shows us how the hydrogens bind. Okay, so like, okay, for NAD, the top one that's written as NADH plus H, you can see the 2 hydrogens, one of them binds right to the molecule, the other one is just loosely attracted to it, so that's why it's called NADH plus H. Down here, FADH2, both hydrogens bind directly to the molecule, so that's why it's considered FADH2. The plus H for NAD there, it just indicates that the hydrogen is not directly bonded to the molecule. It's just, it's loosely attracted to it. That's why. But, you know, again, sometimes you'll see it written as NADH2, but I usually do it as NADH plus H. Okay. So now we're moving into chapter 5, and we're going to be talking about metabolism, which is pretty interesting, I think, because we can really get into some sort of cool topics. Hope we can get through all of them today. Chapter 5 basically talks about glycolysis, which when we look at this word, what do you think it means? Glycolysis. What do you think lysis means? I think you should know this. Breakdown. What do you think glyco means?
Glycogen.
Sugar. Right. Well, glycogen, okay, that's where it does get a little bit confusing because we have a lot of glyco-gluco words here. And then we got lipo. we have a lot of different vocabulary to learn here. And the prefix is always going to be like glyco or gluco, and it gets a little bit confusing. You know, we're talking about sugar, we're talking about glycogen, which actually is a storage form, basically, of sugar, carbohydrate. So glycolysis specifically is the breakdown of sugar, and we're going to talk about the lactic acid pathway and why that pathway sometimes is favored, why it's chosen, and then we'll talk about aerobic respiration, which is where we can really generate a large amount of energy. Lactic acid pathway in humans is not that efficient. We'll be talking about it. And then we'll talk about the metabolism of lipids and proteins. So lipids, we're going to talk a little bit more about than proteins. I'm not going to talk so much about that. But most classes, like for example, if you have to take a nutrition class, you'll see a lot of times they spend a significant amount of time talking about glycolysis. And interestingly, sugar, the glucose is not the preferred energy source of most of the organs of the body. The brain, yes, because we have a blood-brain barrier up there, you know, which prevents a lot of things from getting through. But sugar, glucose, can get through. So that's why the brain definitely uses glucose. But most other organs prefer triglycerides, fatty acids. And so that's why we're going to spend a little bit of time just talking about lipid metabolism. But understanding the glucose metabolism will sort of be a foundation for us. So this is a little bit more specific. The summary, again, like a little mini study guide that I give you at the beginning of each chapter. We're going to talk about the reactions that make up metabolism, anabolism, and catabolism. We're going to describe the events of glycolysis, the Krebs cycle, electron transport chain, and I have actually, when we go on break, I'm going to make some copies. I have a little sort of narrative study guide that will help you to know pretty much the main points of what I would ask you questions about the process of cellular respiration. So I'm going to pass that out to you, and we'll be filling that out together in class. And then we'll describe where the above reactions take place, and the coenzymes, NAD and FAD, we'll be seeing those again. We'll describe the difference between oxidative and substrate level phosphorylation, and if reactions of the above processes are oxidative, reduction, anabolic, catabolic, endergonic or exergonic. So we've talked about endergonic and exergonic and oxidation and reduction, but not yet anabolism and catabolism, which we'll just briefly discuss in a few slides. And then we'll talk about the lactic acid pathway as well, and something called gluconeogenesis, which is really cool because you never think that your body can do this. But interestingly, a lot of these pathways are sort of able to go in one direction, then they can go in another direction. So that's why you'll see like arrows that go in both directions for different purposes. So we're going to talk about that. I think you'll find that a little bit interesting. Okay, so lastly, in terms of coupled reactions, we've got metabolism, which consists of, it's basically the sum total of all of the breakdown reactions and the build-up reactions that happen in the body. Now, specifically, catabolism is where we break down molecules and release energy. This is how, pretty much it's a primary source of energy for making ATP, which is what we need. Anabolism makes larger molecules and requires energy. So like, for example, if we needed to make a hormone like insulin, we needed to make that protein, that would be an example of an anabolic reaction. Or if we needed to make actin and myosin for the sarcomeres in our skeletal muscle, because we're building and we're getting stronger, that would be an example of an anabolic reaction. Catabolism is where we break down things, like for example glucose, like we're going to be talking about, to make energy. So think about it, you know, anabolic steroids, people use anabolic steroids to build muscle. You know, catabolism again is the opposite of that. So let's review briefly the different kinds of reactions that we've talked about in the class total so far, and let's try to categorize them in terms of this example. So we talked about endergonic reactions and exergonic reactions, right? And that specifically refers to our energy transfer. Does our product have more energy or less energy? Then we talked about oxidation and reduction, right? So going from glucose and glucose to maltose, is this going to be oxidative? Is it going to be an oxidation reaction or a reduction reaction? What do you think? If we're making a disaccharide from two monosaccharides, do you think that disaccharide is going to have more or less electrons than the two monosaccharides did? More. You're going to have more because you got a bigger molecule, right? So you've got more atoms, which means naturally you're going to have more electrons. So would the production of maltose be oxidation or reduction?
Reduction.
It's going to be a reduction reaction, right? So, and remember that oxidation and reduction reactions, they're coupled, but specifically they refer to describing whether or not a product has gained or lost electrons. We're talking about electron transfer. So the process of going from maltose to glucose and glucose, is this going to be oxidation or reduction?
Oxidation.
It's going to be oxidation. It's the opposite, right? Oxidation. Okay, and lastly, anabolism and catabolism. Was that your friend?
Yeah.
That was very,
It's
very melodic. Okay, so in terms of anabolism and catabolism, would producing maltose be anabolic or catabolic? Are we building or are we breaking down?
Building.
Right, we're building, so it's going to be anabolism, right? So this is anabolic. And when we break down maltose to glucose molecules, that's going to be
Catabolic.
catabolic, right? So there you go. Now lastly, we talked about one other set of reactions in this class, which may seem like a distant memory for you, but if you recall, we talked about a couple of other reactions, one that was hydrolysis and one that was dehydration. Which one of these reactions is dehydration, the top or the bottom? You guys are on fire. Right, dehydration is the one on the top, because here we're actually synthesizing, we're making a bigger molecule from two smaller ones. So down here, hydrolysis, where we break down a disaccharide into two monosaccharides, makes sense. So this is also a hydrolysis reaction. So you see, we can really use these terms that we just learned for coupled reactions to pretty much describe different things about a reaction. It's just a vocabulary to describe what's happening with a particular reaction. Now with oxidation and reduction reactions, a lot of times we call them redox reactions because that kind of combines the two terms, reduction and oxidation, because they happen together. For anything that gains electrons, something has to have lost them in order for something to gain them. Okay, so again, we're going to see some examples of that as we proceed through this. Do you have any questions so far? Yes?
Are all of those reactions synonymous with one another? Like, is a hydrogen?
They're not really, I wouldn't say they're synonymous because they are describing different aspects about the product that you're producing. So again, you've got endergonic, meaning that your product has more energy. You have the term reduction, which means that your product has more electrons. You see what I'm saying? You're describing different aspects of that product. And that's why I wouldn't say that they're synonymous, but certainly if you're trying to figure out if you've made a bigger or smaller molecule, if you hear one of those terms, then you would know if it's bigger or smaller. But again, they describe different things. It's a vocabulary to describe. It's sort of like, how can I say, like, okay, there's a tree outside. I see a tree. It's got brown bark. It has green leaves. It has leaves that have a particular pattern on them. You can't really use those descriptive terms interchangeably
Okay.
to describe that. Like you can't say brown and then you've got the leaf, you know, and describe the leaf that way. You can't.
I get it.
So that's what I mean. It's just they're not synonymous, but they're describing the same thing, but it's just different aspects of that thing. Yes.
What's the word underneath oxygen? I can't see.
Oh, exergonic.
Oh.
Exergonic. Sorry about that. So one downside of this room is that the screen takes up so much of the board and then you can't see. If you guys need to come up here or you need to like move around, feel free. Anyone else have any questions or can't see or have a question about what I wrote? Can't read my writing? It's possible. All right, so if no questions, then let's move into metabolism. So this is a general diagram showing us very basically what we're going to be describing. So if we eat a piece of bread, for example, and it goes through our digestive system, we know in the small intestine we're going to have some breakdown of this bread, and we're going to get individual glucose molecules which will then be absorbed from the digestive tract into the capillaries that are in those little villi, right, of the small intestine. That glucose goes into the bloodstream and then will make its way into the capillaries, and through diffusion the glucose can make its way into cells that need it to make energy. So glucose will undergo the process of glycolysis, and glycolysis, we'll learn, can occur under aerobic and anaerobic conditions. Aerobic means that oxygen is readily available. Anaerobic means that oxygen is not so abundant. I mean, it's not like maybe you have no oxygen, you just don't have enough. So glycolysis will occur in either of those circumstances, and ultimately we get to pyruvic acid. And pyruvic acid's fate, you know, how it's processed, is determined by whether or not we have enough air or not. So pyruvic acid under anaerobic circumstances will be converted to lactic acid. And that's what in part makes your muscles burn whenever you're doing activity. If there's enough oxygen available, then we go through what's known as cellular respiration, and there are two additional processes. There's the Krebs cycle and electron transport. And we call it cellular respiration because oxygen is readily available. We take in the oxygen, and in the process we make carbon dioxide. Does that make sense? So it's just like how we respire. We take in our oxygen and we exhale carbon dioxide. The cell will take in oxygen and it will respire carbon dioxide. It's the same thing, that's why it's called cellular respiration. But that only happens if you have enough oxygen available. All right, so now let's get into the specifics. Glycolysis. So you don't have to know the overall net equation for glycolysis, because actually I'm going to write this on the board for you, I think in a simpler way. I'm just, at the end of each process we discuss, when we talk about cellular respiration, I'm going to put what goes in and what comes out of the reaction, the major products that you need to know. So you don't have to memorize this part, but it would be beneficial for you to look at this, which is the overall equation for cellular respiration. You start out with C6H12O6, which is what?
Carbonic acid.
No. What does the process of glycolysis do? It breaks down what? What did we say? Breaks down what? Sugar or glucose, right? So C6, remember 1 to 2 to 1 ratio, that's the carbohydrates, right? Glucose, C6H12O6 is glucose plus oxygen gives us carbon dioxide, as we know, cellular respiration produces that, water, and ATP energy. So that equation, I would definitely put a star beside and know that. You never know when that'll come in handy. Okay, so glycolysis itself is exergonic. Okay, the bulk of glycolysis is going to be, you're going to be releasing energy a little at a time. Again, we can't just like break open a glucose molecule and harvest the energy, because if we did that, we would combust. That wouldn't be possible. So we break it down a little bit at a time. If you remember from the enzyme chapter that we looked at, we talked about how there are metabolic pathways. That's what glycolysis and all these pathways are. We have a series of reactions, right? We start with substrate A, we get product B. Product B becomes the substrate for the next reaction and produces product C. C becomes the substrate for the next reaction, and so forth until we get to our final product. And for each reaction that we have an enzyme that helps with the process at each step. And so that's what we're looking at with this process. So we'll see that. Glycolysis is exergonic. It ultimately produces a net of 2 ATP molecules and 2 NADHs, 2 NADH plus H molecules, which are what? Oxidized or reduced? NADH plus H, has it gained electrons? Right, it's gained electrons, so it's reduced. So we have 2 reduced NAD molecules, we could say, 2 NADH plus H. However, glucose first has to be activated by 2 ATPs. We have to get the process started. So initially, with the process of glycolysis, we need to put energy into it. So that means it's endergonic. But ultimately, we break down our glucose and we release energy, and that's exergonic. So the reason why we need to put energy in is because we have a glucose molecule, and we know, as we talked about with cellular transport, when we were talking about our different transport, and I showed you that diagram of absorption or reabsorption. Remember, it's possible if you have a lot of glucose in a cell that through facilitated diffusion, you could lose it, right? So if we don't want to lose the glucose, what we first have to do is we have to change its structure so that it cannot fit into its transport molecule. So facilitated diffusion will not work because glucose doesn't look the same. So how do we do that? We have an ATP molecule, adenosine triphosphate, giving up a phosphate to become adenosine diphosphate, okay? And that phosphate goes to glucose. And when that phosphate goes to glucose, it changes its structure and it can't be transported out of the cell. Now it's trapped inside the cell and it has to be metabolized. Does that make sense? Anyone have any questions about that? Then the second ATP that's needed is going to donate a phosphate to our new molecule to basically make it symmetrical. So we're going to see that, and I'll draw it on the board for you. And then once we get to that level, and notice here too, you know, the free energy level of glucose goes up because it becomes a larger molecule. So that's why you see the free energy first increasing, and then the exergonic part occurs, where we have the breakdown of what we've created, and in the process we produce ATP and we produce 2 NADH plus H molecules. So that's exergonic. Okay, so initially it's endergonic because we have to put 2 ATP in, and then ultimately it's an exergonic. And then we produce in the end pyruvic acid. That's our final product in this metabolic pathway. Do you have any questions right now? All right, so this is the big time slide, and everybody's like, whoa, it looks so complicated. Honestly, you don't have to know every single product that's produced here. This is a metabolic pathway, like I said. We start with the first substrate or reactant, which is glucose. Then we produce a product. Then we have that product becoming the substrate for the next reaction. And then we produce a new product, which becomes a substrate for the next, and so forth. And this continues until we get to pyruvic acid, which is our final product. Okay? And in between where you see these arrows, there's an enzyme that's helping to get these reactions moving more quickly or catalyzing the reactions. Does that make sense? Okay, so I will write for you exactly what you need to know. But first of all, what I want to explain to you is this idea, what happens with the glucose molecule initially. Because I know it's a little bit perplexing, wondering, you know, why do you have to put ATP into this? Well, glucose, okay, has 6 carbons. We know C6H12O6. I'm going to draw my carbons as little circles. So 6 carbons. Okay. And in chemistry, we can label our carbons with numbers. So we have 1, 2, 3, 4, 5, 6. So we have glucose, our first ATP molecule donates its phosphate to make something called glucose 6-phosphate, which means that the phosphate from ATP is going to be attached to the 6th carbon of glucose, okay? And then we have another reaction that occurs, okay? So when we add that, then we have glucose 6-phosphate, then another reaction happens, and then it becomes fructose 6-phosphate, which is a different molecule altogether. And then ultimately the second ATP is going to donate its phosphate to the fructose molecule, the fructose 6-phosphate, to become fructose 1,6-biphosphate, which means that now the molecule has 2 phosphates. That's where the biphosphate comes in. And the 1,6 means that the phosphates are on the 1st and the 6th carbons. So this is fructose 1,6-biphosphate. Now, what's the purpose of that second phosphate? It makes the molecule symmetrical, okay? Because then the very next step, what we do is we cut the molecule in half. We cut it into 2 3-carbon molecules. Like that. You see, so they're symmetrical. And then there's a series of reactions that ultimately gets us to our final product. They're processed exactly the same way. Does that make sense? No? You gotta watch the video again. All right, so what doesn't make sense? What would you like for me to explain again?
Nothing. I just have to read it in the book, I think.
I don't know if reading it in a book would be better. Here, I'll explain it again. So, first we start with the glucose molecule. We need to first add a phosphate to our glucose molecule to change its structure so it can't be transported out of the cell. So our first thing that we make is glucose 6-phosphate, which means that our phosphate is first added to the 6th carbon. Then the second ATP is going to donate its phosphate to make the molecule symmetrical. So we add it to the 1st carbon, and then in the next step, it's split into 2. So you have 2 3-carbon molecules which are symmetrical, and then ultimately they can be processed exactly the same way. And they produce the same products on each side. That's the whole idea.
So like just like an equation, it has to be balanced on both sides to split.
Yeah, it makes it more efficient that way. Yeah. Yes?
Okay, sorry, but you go too fast. So step 1, you start with a glucose molecule.
Mm-hmm.
Step 2, the point is to add a phosphate to a glucose molecule.
Right.
Is step 3,
Well, step 1 is to trap the glucose inside the cell. That's why you add the phosphate. That's important to understand. Then the next phosphate is added to make the molecule symmetrical so that when it's broken in half, both sides of the molecule can be processed exactly the same way on this side and this side.
symmetrical.
You need me to repeat something? Huh?
Like the whole thing. I got 2 broken in half.
Yeah, and then they
That's
can
like
be
the,
processed the same way on both sides. So here's what I'm gonna do. I'm going to break this down for you in a simple way so that you understand the main points to this metabolic pathway that I want you to know, okay? And then we're gonna take a break. I'm gonna grab my handouts and then make copies, and then we'll come back and we'll go through the handout together, so we'll repeat it again. So glycolysis, which is the breakdown of sugar, what goes in and what comes out of this process. Okay, first we have 1 glucose molecule. What comes out ultimately are 2 pyruvic acid molecules. If you see, we start here with the glucose, we end up with 2 pyruvic acid molecules down here. We also have 2 NAD molecules plus 4 hydrogens, and we get 2 NADH plus H molecules that are produced, 2 reduced coenzyme carriers. Why did I say we have 4 hydrogens? Because each of the NAD molecules will hold 2, right? And we have 2 NADs, so we can take on 4 hydrogens. That's why I put that. And lastly, we actually had 2 ATP that went in, and have 2 ATP which come out. Now I want to explain that a little bit better. So when you look here, how many ATP do you see that are produced? Remember, the arrow shows you what you're producing. So whatever's at the tip of the arrow, you're producing. Okay, we have 1 ATP here and another one here. And of course, our molecules are symmetrical, so the other side will yield the same. We have 1 ATP here and 1 ATP here for a total of 4, right? But we needed to put 2 ATP into this reaction to begin with, right? So essentially, you've only netted 2. Even though you've had 4 ATP produced, you basically lost 2 because you had to use them in the beginning of the reaction. So you've only generated 2 ATP. So 2 ATP net is what you've produced. So this is pretty much it for glycolysis. If you know these numbers and you know what goes in, what goes out, you're pretty good with that. And then understanding the purpose of the first ATP and that phosphate to generate glucose 6-phosphate is also important, because we're going to see when we talk about lactic acid production where that is important that you understand, because there's an enzyme we're going to talk about that actually has the name glucose 6-phosphatase. And so understanding that little concept there is going to help you to understand the reaction of glucose 6-phosphatase when we get back. Any other questions? I'll go make some copies of that handout, and I guess come back at 10:42.